day, grade 12 learners. Welcome to today's Life Sciences lesson. My name is Fawzi Rahman. This lesson is brought to you by the Gauteng Department of Education in collaboration with Saibono Discovery Center. My colleague Radina Kruger and I are going to do the final push revision for the final exam. Right, so please pay careful attention. Okay, I think I'm stuck. Just give us a few minutes to get this going.
The road to success is to commit to yourself. In order for you to become who you want to become as a student, you need to commit to reading your books, to studying, to bettering yourself. I know it's not going to be easy. Actually, no one said it's going to be easy. But if you put in the work, the results will show in your life. Tap into your power. Sometimes you will feel like putting away your books, and not reading as much as you're supposed to be. But I want you to commit, because through commitment, there is infinite power.
Right, welcome back. Sorry about that. So as I was saying, we are doing the final push revision and we are addressing the problematic areas from the life sciences prelim exam. So we have discussed, this is the structure of the paper. You will see there's human reproduction for 41 marks, responding to the environment 54, and the endocrine system 34. These were the problematic areas that we found in the prelim exam. Right, so we're going to be discussing those topics. And if you look here, this is the structure of your question paper. So please note that this is the paper 1 and paper 2 is the same, but anyway, we have 150 marks for 150 minutes. So what you need to note is that in life sciences, it's 1 mark per minute. Section A is the short questions for 50 marks here. And then there's section B with question 2 and question 3, 50 marks each. So this should give you a guide onto how much of time to spend on these questions. Excuse me. Right, so you should spend less than 50 minutes on section A, so you have more time for question 2 and for question 3. At about 10:30, you should start off with your question 3. You can leave it at question 2, go back later on after you finish question 3 to give yourself enough time to read over the paper and answer all the questions. You can always go back to what you've left out. Right, we're looking at human reproduction, and this is the exam guidelines, which you must have with you, which tells you what you need to know for the exams. Right, you need to know the female reproductive system, and you must know the diagram of the ovum. Then it's the male reproductive system and the diagram of the sperm cell. So make sure you know how to draw these, put labels, and you must have a title. Right, we're going to discuss a little bit on puberty, and then we're going to talk about the menstrual cycle, hormonal control, and then development of the fetus. Right, let's get into it. This is the male reproductive system. Now, the male reproductive system consists of a scrotum. There's it here, right? The scrotum is a pair of testes which is found inside the scrotum, right? The scrotum is a sac-like structure and the testes are found inside the scrotum. Then you have the structure here, which is the epididymis, right? The vas deferens here, which is also called the sperm duct. And then once it passes the ureter on this side here, it's called the ejaculatory duct. And then we have the urethra here, right? The urethra is found in the penis, and that is the path for semen and for urine. Then we have accessory glands. They are the seminal vesicles. These ones here are the seminal vesicles. And we have the prostate gland here, and then we have the Cowper's gland on either side here as well. So you need to know the front view and the side view. So if you look at the side view, you find the seminal vesicles here. This is the prostate gland here, right? The Cowper's gland is, you can't see it on this diagram clearly. There's the urethra, the vas deferens, right? The bladder, the epididymis, the testicles, which is housed in the sac-like structure called the scrotum. Now, we are going to look at the accessory glands. The seminal vesicles, they produce fluid to transport the sperm cells, and they produce fructose for nourishment of the sperm. Then we have the prostate gland, which secretes a milky alkaline fluid that neutralizes the acidity of the vagina. It has enzymes and nutrients for sperm. The Cowper's gland secretes fluid for greater motility of the sperm. So you need to remember that the prostate gland and the seminal vesicles, they provide nutrients for the sperm. Right, we're going to do an activity from Gauteng Prelim Paper 1 this year, 2022. Let's look at the diagram quickly. It says, give the letters of all the structures that are involved in each of the following. So we want the letters of all the structures and we want letters. Please make sure you read the stem of the question and you read the question to understand what is required of you. Right, so if you look at this, production of the fluid part of the semen, right? That's what you want. You want letters for production of the fluid part of the semen. Right, so you will not say D, because D now is where the sperm is being released. But remember, semen is made up of sperm and fluid that comes from the prostate gland. So we'll have B, that comes from the seminal vesicle, we'll have A, and coming from the Cowper's gland, which is then C. So A, B, C, and 1 mark for all 3 of those, otherwise no marks. Right, which is the passageway of sperm and semen, and we spoke about that, and we said that's the urethra, but we want the letter here, so that's E. And name gland B, right? Gland B, we spoke about the prostate gland. Now, the hormone produced by the testes that brings about development of male secondary sexual characteristics. Which hormone is that, learners? This is the one that you should know and very well, and that is testosterone. Right. The next question is, test results show that this man has a low sperm count. Explain why a doctor would advise him to wear against wearing tight underwear. So why mustn't he wear tight underwear? It's the same question that you can be asked in different ways. So we could say, why must a male not take hot showers? Why must he not sit in one spot all the time? So the answer would be the same. So everything you are going to be tested on in the exam comes from the exam guidelines, and you have been through it with your teacher. You need to adapt, think about the work that you've done and adapt it to the question that we ask you. Right, so let's see the answer for this. Tight underwear tight underwear will cause the testes to be close to the body. The temperature of the testes will then be too high, and sperm will not mature, or there'll be no sperm production, or sperm will be negatively affected. It is very, very important that you don't say sperm will be affected. You need to say how in life sciences. So you need to say negatively, negatively affected, right? Now, number 1.4, explain what would happen to the male's ability to reproduce if part label D is blocked off. So there's part label D, and we're looking here at the sperm duct or the vas deferens. And if this was blocked off, right, what would happen? So I could ask you the same thing in a different way. We could talk to you about a vasectomy, and the answer would be the same, right? No sperm will be transported to the penis, which will result in no fertilization, and no offspring will be produced, or there'll be no reproduction, right? So we're talking here about the ability to reproduce. Therefore, you need to say no reproduction, no offspring, or no fertilization will occur, because no sperm will leave past this point if it is blocked or it is cut off by a vasectomy. Right, the female reproductive system. The female reproductive system is made up of the ovaries, one on either side. So there's a pair of ovaries, the fallopian tubes, the uterus, right, and the vagina. So if you look at the uterus here, the uterus is for protection. It looks after the embryo. Then we look at the fallopian tube. The fallopian tube transports the ovum to the uterus. It's also the site for fertilization, right? And then we have the ovaries on either side, and the ovaries produce the sex hormone, which is estrogen. Now remember, ovaries are glands because they produce hormones, and the testes are also glands because they produce the hormone testosterone. If you look at the side view, you find the urethra there, and there is the cervix here, and this is the cervix here. This is an important part. It is the mouth or the opening of the uterus, right? And you can see here, you have your fallopian tube and there's your ovary there. Right, so let's look at an activity. The diagram below shows the female reproductive system. Identify part A. So we've spoken about part A. That one is the fallopian tube. And then C, we've spoken about C, which is the cervix or the mouth or the opening of the uterus. But you need to show us that you've been through the life sciences course and you need to say the proper terminology, which is cervix. Give the letter only. Give the letter only. Take a highlighter into your exam and highlight on your exam paper. Give the letter only of the part where the following takes place. Meiosis. Meiosis will take place and then gametes are produced. So that should be in the ovary, which is B, and fertilization will be in the fallopian tube, as we said, and that would be part A. State two functions of part E. Right, what is part E? If you look here, part E is the uterus. Now, this is the uterine wall, right? That is the uterine wall. And this lining here in red that I've drawn, that is the endometrium. So we want the function of part E, which is the uterus. It's responsible for the protection of the embryo from implantation to birth, and it provides space for the developing fetus. Name the glands which secrete nutrient-rich fluid for the sperm to reach part A. Now, this gland is not anywhere on this picture, right? We want you to look at, or to name rather, the glands which secrete nutrient-rich fluids for the sperm to reach part A. So now we are talking about the male reproductive system and the nutrients. Remember, we spoke about the prostate gland and the seminal vesicles. Right, we're going to move on to puberty. So puberty is when you're not a child and you're not an adult. So it is a confusing stage for lots of you. And in males, it is caused by it's caused by the hormone testosterone. And in females, it is the estrogen that is being produced by the follicles. So in females, the ovaries start producing the hormone estrogen under the influence of the follicle stimulating hormone. Now, when you are born, you have follicle stimulating hormone and luteinizing hormone and all these cells to produce ova that you will ever have for the rest of your life. And then it decreases, and when you are undergoing puberty, then it starts increasing again in concentration, and that's when the follicle stimulating hormone increases, and that's when it will produce the follicles, which will produce the estrogen. And then what these hormones do, they help you to develop secondary sexual characteristics, which change rather a girl into a woman and a boy into a man. So you will have hair on your face if you're a man, your shoulders will be more defined. If you're a female, you will have breasts will develop and armpit hair and so forth. Right. Another thing that happens during puberty is there is formation of gametes, right? So let's look now at gametogenesis, which is the formation of gametes. In males, you have the formation of the sperm cells, and in females, you have the ova, right? It's one ovum, many ova. Please refrain from using the word egg or egg cell. As I've told you, we need to see that you've been through the life sciences course. You need to use correct terminology. So gametogenesis is the formation of gametes. And how are gametes formed? They are formed by the process of meiosis. In males, the gametes are the sperm cells, and the meiosis in males is called spermatogenesis. In females, ova are produced, and that is caused by meiosis in females, which is, the name of it is oogenesis. Right, now remember when we looked at the exam guidelines, we said that you have to know the diagram of the ovum. So please make sure you learn to draw the ovum. It consists of a haploid nucleus, right? And it has cytoplasm, and then it has a cell membrane, and it has a jelly layer. So make sure you learn how to draw this diagram. Right, in the males, we have the sperm cell, and you need to learn how to draw the sperm cell. Right, the sperm is made up of a head, which has a nucleus, And this nucleus has a haploid number of chromosomes. So the head is made up of the nucleus containing 22 autosomes, and the 23rd sex chromosome, or we prefer the word gonosome, is either an X or a Y. Remember that the sperm only has a haploid number of chromosomes. So you have 22 autosomes, and the 23rd gonosome is either an X or a Y. If it is a Y and it fuses with the X, then that will be a male. And if it is X and fuses with the female ovum, the females are only X. We can only be X and X. We can only make females. But the male determines the sex of the offspring. Right, then you have the middle piece, which is has mitochondria to provide energy for locomotion. And then we have the acrosome at the head, which contains enzymes that dissolve the ovum's membrane during fertilization. And we have a long tail here to help the sperm to swim. Remember, the sperm is a tiny, tiny cell, and the ovum is one of the largest cells. So it has a long distance to travel to fuse with the ovum, and therefore it needs a tail and it needs energy, which it obtains from the mitochondria. Right, so let's look at the Gauteng prelim paper again, and we're going to look at the structure and how is the following structures suited to assist the sperm cell to perform its function. So now we're not asking you what gives the cell energy. We are saying, how does this assist the cell, the sperm cell, to perform its function? We've spoken about the mitochondria. There's increased surface area for cellular respiration to provide energy, and the acrosome has enzymes to dissolve the outer membrane of the ovum for the sperm cell, the nucleus of the sperm cell, to enter the ovum for fertilization. Right, sperm cells and ova are produced by gametogenesis. This happens when diploid cells undergo meiosis to produce haploid cells called gametes. Tabulate two differences between gametogenesis in males and gametogenesis in females, not mentioned above. So there's a table. First of all, it says tabulate, and when it says tabulate, it means draw a table. Right, and it's 1 mark just to draw this table. Then gametogenesis in males is called spermatogenesis. In females, it's called oogenesis. It's stimulated by testosterone in males, and it's stimulated by the follicle stimulating hormone, or FSH, in females. It takes place in the testes, in the seminiferous tubules, in particular in males. In females, it takes place in the ovaries, in particular in the follicles. Gametogenesis results in 4 sperm cells being produced in males and 1 functional ovum in females. It starts off in puberty in males, and the process starts at birth in females. Right, we're now going to look at the menstrual cycle. This is a graphical representation of the menstrual cycle. And remember, the menstrual cycle is made up of the ovarian cycle and the uterine cycle, okay? So if you look here, we have the ovarian cycle. This is what is taking place in the ovary, right? Then we have the pituitary hormones. These hormones are secreted by the pituitary gland. We have the ovarian hormones, and we have the uterine cycle. This is what's happening in the uterus. Right, if you look now from day 1, and this is your time here, right? This is your timeline at the bottom here, and it's showing you the number of days. You may not get an exact graph like this in the exam. You may get something different, but if you know what is happening on each day, you can apply this knowledge to the diagram that is given to you or the situation that is given to you in the exam, and you will be fine. Right, so the pituitary gland, remember, is secreting the hormone FSH. So what does FSH do? The name will tell you, follicle stimulating hormone. It stimulates follicles to be formed. So there's your follicles here that are growing. Okay, there's your follicles. Now, from day 1 to 7, from the previous cycle, what happens? It is menstruation. Now, you need to remember that menstruation is a process. The menstrual cycle is a cycle made up of the ovarian and the uterine cycles. The follicle is a structure. So when we ask you to give a structure, give a process, name the cycle, you need to be clear in what you are going to answer and not give us the wrong thing. Right. So from the previous cycle, you find that this person is still undergoing menstruation, and you can see that because there's a decrease in the endometrium, right? So that process of menstruation is occurring here. That's day 0 to approximately day 7. All right, what else is happening? The follicles are growing. When the follicles are growing, stimulated by the follicle stimulating hormone, the follicles are producing the hormone estrogen. So you can see here that estrogen is slightly increasing now because these follicles, as they're growing, as they're maturing, they're producing the hormone estrogen. And the follicle stimulating hormone is the light blue one here. You can see it's a little bit high because it is stimulating the follicles to be formed. Right, then we move from day 8 here to day 13 there, okay? The ovaries here are producing, these follicles are growing in the ovaries, and in this ovary, the Graafian follicle, one of the follicles, mature and is called the Graafian follicle, right? It secretes estrogen and Let me stop there. What is the function of estrogen? Guys, you have to know the function of estrogen, right? Estrogen stimulates this endometrium to become thicker, to become more vascular, and to become glandular. So the function of estrogen is to build the endometrium. So here you can see as the estrogen level is getting higher, you can see that the endometrium is getting thicker. It's being built. So when we talk about more vascular, we mean that there's more blood in the blood vessels. When we say more glandular, the glands have more mucus in them and they become thicker. That makes the endometrium become thicker. Right. So this is what happens. That's the function of estrogen. So from day 8 to day 13, this is what's happening. The endometrium is being built by the hormone estrogen. And then just before day 14, around about day 13, you can see here that estrogen is at a very high level compared to anywhere else on the graph. This high level of estrogen, it will stimulate the pituitary gland to produce the luteinizing hormone, right? And what is the function of the luteinizing hormone? The function of the luteinizing hormone is for ovulation, the process of ovulation. It stimulates the process of ovulation. Ovulation is when the Graafian follicle bursts open and expels an ovum, as you can see there, right? The Graafian follicle will burst open, expel the ovum, and then the second function of the luteinizing hormone is to make this empty Graafian follicle after it expels the ovum to form this structure here called the corpus luteum. Right. So now that's what's happening. So day 13, there's a large amount of estrogen, the luteinizing hormone is high, and ovulation will occur on day 14. Right? So in the middle of the menstrual cycle, which is day 14, the process of ovulation occurs where the Graafian follicle bursts open and releases that ovum. Right, now the corpus luteum forms by the influence of the luteinizing hormone. The function of corpus luteum, it produces the hormone progesterone. So now if you look here, you can see this, the dark blue graph here, it's increasing because now corpus luteum is producing progesterone. So before I move on, let's talk about progesterone. Progesterone is a hormone, and I know many of you remember it. You say P for progesterone, P for pregnancy, but you need to explain what progesterone does and the function of progesterone. Progesterone keeps the endometrium thick, makes it a little bit thicker, in fact, more vascular, more glandular, and keeps the endometrium, maintains the endometrium as well in case a pregnancy should occur or a fertilization takes place, then the endometrium acts as a cushion for the implantation of the embryo. All right, so this is what's happening. The progesterone level here is increasing. Now you can see here that estrogen increases slightly as well, but that is because corpus luteum secretes a tiny amount of estrogen as well. But you need to worry about the corpus luteum and the progesterone. So can you see now how the endometrium is thick, right? It's thicker, it's vascular, it's glandular, in case a fertilization occurs. Now, when we look, we've spoken about day 14, and now when we look here around day 28, when you see that the progesterone level is decreasing because the corpus luteum is shrinking and disintegrating because no fertilization has probably occurred. Because there's no fertilization, the corpus luteum will shrink and therefore it will produce less progesterone and the progesterone level decreases. And when the progesterone level decreases, this has an effect on the endometrium. The endometrium will break down and will be released as menstruation. Okay, right, the body temperature, I want to speak about this. You can see that during ovulation, the body temperature increases slightly, right? And then it goes back to normal temperature again. And some people even use body temperature as a means of contraception. So they know that during the time of ovulation, it is their fertile time. Okay, I think I've covered it there. One more thing I want to mention, learners, is that if you look at the follicle stimulating hormone, right? The follicle stimulating hormone is high, relatively high when the follicles are growing, right? And you can see that progesterone is low. But here, right, from day 15 to day about 23, you can see that the progesterone level is high. But the follicle stimulating hormone, the light blue one here and the dark blue one in this graph here, in this section, it's a dark blue. Here, it's a light blue. The follicle stimulating hormone is low. So in other words, this is a negative feedback mechanism. The follicle stimulating hormone inhibits progesterone, and when progesterone is high, it inhibits the secretion of the follicle stimulating hormone. So that is the negative feedback mechanism. So you can see here, the follicle stimulating hormone is low, progesterone is high. No more follicles need to be produced because in case there is a fertilization that occurs, right? So our body saves our hormones. It takes energy to make hormones, and our body saves that energy by producing large amount of hormones when we require them. So we have all the hormones in our bodies at most of the time in our cycle, but at certain times, a hormone will be more than at other times. Now, we're going to look at if fertilization takes place. So the first part of the graph is the same as the previous one, but if fertilization takes place, then the second half is different, because what happens is the corpus luteum now, it does not shrink, it does not disintegrate, and it does not stop producing progesterone. It continues producing progesterone, right? And you can see here that the progesterone level is high, and we need to maintain this endometrium for the implantation to occur, or for the fetus then to grow and form the extra embryonic membranes to look after that developing fetus. So you can see here that from day 28, after day 28 onwards, we're going back to the beginning of the cycle, and there is no menstruation occurring. The endometrium is still built up, it's still high, it's being maintained by progesterone. So progesterone maintains the endometrium, thereby maintaining pregnancy. Don't just say the progesterone maintains pregnancy. We need to know how pregnancy is maintained. So you got to speak about the endometrium. I'm going to go through this very quickly. This is a quick summary of what I've spoken about already. There's a pituitary gland secreting the hormone FSH there, which stimulates the development of the follicle in the ovary, which produces the hormone estrogen for the O, and this causes the thickening of the endometrium. Then we have pituitary gland producing the luteinizing hormone at the time of ovulation, which causes ovulation and the corpus luteum to form. And the function of corpus luteum is to produce the hormone progesterone, which maintains the endometrium, thereby maintaining pregnancy should it occur. Now, should a pregnancy occur, right? Let's talk about a fertilization firstly. So let's look at this diagram, right? This diagram shows you here, there's the ovary, and the ovary is secreting an ovum, right? Or producing an ovum rather. And then this box here is magnified and showing you fertilization here. So what is fertilization? Fertilization is when the haploid nucleus of the sperm cell fuses with the haploid nucleus of the ovum, right? And you can see here that all the sperms are going for this ovum, but only one will penetrate this ovum, and then a hard covering forms over the ovum, and no other sperm can penetrate that ovum. Now, what happens? This continues moving along the fallopian tube to form a zygote. A diploid zygote forms once the nucleus of the sperm fuses with the nucleus of the ovum, right? That is fertilization. Once that occurs, a diploid zygote forms. And that is one cell, but it has 46 chromosomes in it for the human, right? Then it continues to grow by a process of mitosis, right? And by the time mitosis will occur until this zygote becomes an adult, where you have more than 3 trillion cells once you reach an adult. But now, this process is continuing, and then you come here to what is called a morula, because it looks like a mulberry. It's a whole cluster of cells. And then you get the blastula, right, or blastocyst. The blastula has fluid in here. It's a fluid-filled structure with cells, and this moves along. There's it here. It moves along the uterus and implants into the endometrium. And when this implants into the endometrium, the process is called implantation. And then only you can say that this female is pregnant. Only when implantation occurs and you undergo a test will we say that this female is pregnant. Now, the development of the fetus. As the fetus is growing, you can see here, this is the amnion, right? It is let's start off with the chorion as the outer layer here. Then you get the inner layer, which is called the amnion. And the amnion gives rise to the amniotic fluid inside here. And this is the amniotic sac. Then you have the umbilical cord, which is made up of the umbilical vein and the umbilical artery. And the umbilical vein, right, and the umbilical artery. The artery takes waste away from the fetus to the placenta to be diffused into the mother to be excreted, and the vein brings nutrients to the developing fetus. Now, the chorion, together with the chorionic villi and the endometrium, form the placenta, right? So there are spaces in here in the endometrium where the chorionic villi comes together with the endometrium, and the mother's blood is close to the blood from the fetus. They come close together like this, but they do not mix. Okay, the fetal blood and the blood of the mother do not mix. And you need to remember as well that the chorion forms the chorionic villi together with the endometrium, which forms part of the placenta. Now, the amniotic fluid. The amniotic fluid acts as a shock absorber. So if the mother has to fall, it keeps the fetus hydrated. It prevents it from desiccation and dehydration. It acts as a shock absorber, right? And it allows the fetus to grow into a baby. So this is a summary of everything I have spoken about. And I'm not going to spend too much of time on this because I have spoken to you about everything that is on this diagram. Besides the placenta, it allows for gas exchange to occur, providing nutrients to the fetus. And the chorion is the outermost extra embryonic membrane covering the amnion, and it, together with the villi, it forms the placenta. Right? We spoke about the umbilical arteries carrying waste, carbon dioxide, and metabolic waste to the placenta to be diffused into the mother. and from the mother, nutrients are diffused into the developing fetus. Right. Okay, we're going to look quickly at some questions. Now, investigations were a problem in the prelim exam, and we have one here on the section, so we're going to look at this quickly. An investigation was conducted to determine the effect of zinc supplements on the level of testosterone in the blood of weightlifting males, right? 60 healthy weightlifting males of the same age were asked to participate in the investigation. They were given the same diet for 6 weeks. The testosterone levels in the blood was measured every day for 6 weeks. They were given a zinc supplement with their diet every day for 6 weeks. The testosterone levels in the blood was measured every day. The average levels of testosterone in the blood was calculated before and after the zinc supplement was given. So before the supplement was given and after the supplement was given, the testosterone levels were measured. If you look at this table, it's a summary of what is happening. Before administering the zinc supplement, the testosterone level in the blood was 16.8 micro per milliliters, right? And after administering the zinc, it shot up to 22.5. So we can see clearly here that there's a relationship. And there's a relationship between the zinc after administering the zinc. So the zinc supplement has an influence on the testosterone levels. So when you are given a diagram or you're given a table, unpack this table. Look at what it's saying. See what is happening. Are the numbers increasing? Are they decreasing? What is the relationship between the two? Okay, so you need to look at that. Right, now I want to go back. Right, the independent variable is the zinc supplement and the dependent variable is the testosterone levels in the blood. I'm just trying to go back. I want to show you where I got these two from, right? So identify the independent variable. The independent variable and the dependent variable you get from the aim of the investigation. Guys, it is no more looking at the table and saying the first column, that is the independent variable, that's the x-axis. No, you need to look at the aim of the investigation. An investigation was conducted to determine the effect of zinc supplements, to find out, okay? So that is the aim of your experiment, and that is where you get your dependent and independent variables from. The effect of the zinc supplement on the levels of testosterone. So there you get your independent variable and your dependent variable. So your independent variable then is your zinc supplement and your dependent variable then is the testosterone levels in the blood. Describe the control. Remember we said the zinc supplement was testosterone levels were measured before and after the zinc supplement. So that would be your control. Right? State 2 variables that should have been considered about the zinc supplement specifically. So you can look at the type of the zinc product, the concentration, volume of zinc, time that the zinc was being administered, and the way the zinc was administered as well. State 2 ways in which reliability of the results were ensured, and that is because 60 males were used, so it was a large sample, and also the time where the period was over 6 weeks. Write a general conclusion, and remember I told you that you saw that the testosterone levels increased as we gave the zinc supplement. So zinc supplements increase the testosterone levels. It's 2 marks here or nothing. Give a statement for the conclusion where you speak about the relationship between the two variables. State 2 functions of testosterone in male reproduction. It stimulates the production of sperm cells and it stimulates puberty. Right. So if you look at, because of time, I'm going to move on to the next section. And the next section that we are going to discuss is the endocrine system, which was not answered well in the exam as well. Right, so the endocrine system and homeostasis. You need to know your glands and you need to know the hormones that those glands produce when you're looking at the endocrine system, and negative feedback mechanism, what that involves, and homeostasis, which is an internal balance of your body. And particularly you need to look at how glucose is balanced, carbon dioxide, water, and salts. Obviously, we're not going to have time to go through all of this, but you need to go through all of this because it's in the exam guidelines. And even though I may just do one or two from here, any one of them can come out in the exam. The next one that's very, very poorly answered in paper 1 is thermoregulation. So we're going to try and look at these quickly. Right, the human endocrine system, right? We have, so the endocrine system is a chemical system. It's a slower system compared to your nervous system, right? There is communication. So your hormones, it's dealing with hormones. Your hormones are produced in one part of the body. Those hormones are they then travel in the blood and they come to a target organ. And then when the target organ realizes, I have this amount of hormone in my blood, therefore I need to perform this function. I need to do this. I need to do that. So that's what happens in the endocrine system. It is the hormone part of your system, of your body. It works slowly because remember the hormones are traveling in the blood. So I'm going to look at this quickly. You have the hypothalamus, which produces the antidiuretic hormone. And then you have the pituitary gland, which is the master gland of the body. Why is it the master gland? It's this tiny pea-sized gland here, this tiny gland here, right? Why is it the master gland of the body? Because it stimulates other glands to produce hormones. Remember, I said that the testes and the ovaries are also glands, so they produce hormones that is also stimulated by the pituitary gland. So you see that here we have TSH, which is a thyroid stimulating hormone that stimulates the thyroid gland. FSH, remember we spoke about that, the luteinizing hormone, and prolactin, which is for milk production. Then you look at your kidneys, and just above your kidneys, you have these glands here. There's 2 of them above each of your kidney, and those are called the adrenal glands, and they produce 2 hormones, adrenaline and aldosterone. Now, you need to learn what adrenaline does. It increases your heart rate, raises your blood pressure, causes pupils to dilate. It's known as the flight or fright hormone, which is secreted when you are scared. And examiners usually couple or join this hormone together with the nervous system. So make sure you learn everything about adrenaline. And then we look at aldosterone during homeostasis, because this regulates the amount of salt in the body, and also it works together with the antidiuretic hormone to bring about water balance or osmoregulation. Right, the testes and the ovaries we spoke about. The testes producing testosterone, therefore it's a gland, it produces a hormone, and the ovaries with estrogen and progesterone, therefore it's a gland as well. Now we're looking at the pancreas. Look at this green tongue-shaped structure here. It's found slightly below the stomach. It is called the pancreas. It secretes 2 important hormones, right? Insulin and glucagon. Now, learners, you need to learn the spelling of glucose, glycogen, glucagon, because in the exam, for glycogen, if you say glucogen, for example, we're going to mark it wrong. Because we are going to say, you are not sure whether you want to write glucose or you want to write glycogen. So we will mark that wrong. You must get the spelling of these correct. So it means you need to learn these properly. Right, what does insulin do? It reduces the blood glucose level. So when you, after a meal, your blood glucose level goes up high, insulin is then released by the pancreas to then lower the glucose level. How is that done? Glucose is converted into glycogen, which is stored in the liver. Glycogen is insoluble. Then we have glucagon, which performs opposite to insulin. What does glucagon do? It raises the blood sugar level by converting the stored glycogen into glucose. Then we have thyroxine, right? There is the butterfly gland here at the top of your esophagus and your throat is called the thyroid glands. There's 2 of them on one of them on either side. It regulates your metabolic rate. It affects the growth and development and functioning of your heart and your nervous system. right? So make sure you know about the thyroid gland as well. Right. Now, what is the negative feedback mechanism? As I was telling you, it's a sequence of about 7 steps where there's an imbalance that is detected in your hormonal levels in your body, and a control center is then stimulated. It then responds. In step 4, a message is sent to the target organ, and the target organ will then respond and reverse the balance to obtain then normality, right? So balance is restored in a negative feedback mechanism. And we're going to look at examples of this, right? It's made up of 3 main components. There's a receptor, there's a control center, and an effector. So if you look at this guy in the middle, this is normal, right? He is normal. And then when his body temperature decreases here to the left, you find that he starts shivering, and that helps him to that increases the body temperature and brings it back to normal. If his body temperature increases, then a cooling process occurs by sweating, and then that decreases the body temperature and brings that level to normal. Right, so we're going to look at the regulation of the thyroid levels in the human body, right? And the first thing we're going to look at is high levels. So let's see, step 1. When your thyroxine levels in your blood increase above normal limits, then the it's traveling, remember there's thyroxine, it's traveling in your blood, and it goes to your pituitary gland, and the pituitary gland senses that there is too much of thyroxine in the blood. So what is the pituitary gland going to do? It's going to secrete the thyroid stimulating hormone. It's going to produce less of the thyroid stimulating hormone because there's too much of thyroxine. So can you see the negative feedback here? There's too much of thyroxine, therefore less of the thyroid stimulating hormone will be produced. So that is what we call negative feedback mechanism, where too much stimulates less of another hormone, and when there's too little, it stimulates more of another hormone. So that is the negative feedback mechanism. Right. So we said that the thyroxine levels in the blood are above normal. The pituitary gland is then stimulated to produce less thyroid stimulating hormone. As this moves along in the blood and the blood reaches the thyroid gland is going to sense that there is less thyroid stimulating hormone. Therefore, it produces less thyroxine, and that level is brought to normal. Right. Now, when we have low levels, the opposite is going to happen, right? When the thyroxine levels in the blood is below normal, the pituitary gland is stimulated to produce more TSH. So can you see the negative feedback mechanism here? High TSH levels stimulate the thyroid gland, and it produces more thyroxine, and the level of thyroxine increases and therefore goes back to normal, right? And this is just a flow diagram on the side here to show you the same thing. Now, sometimes a very important micronutrient is required to look after to look after the thyroid gland, and that micronutrient is called iodine, right? And when there's a lack of iodine in your diet, then the thyroid gland swells and looks like this diagram that we have here, and there's coughing and hoarseness and changes in your voice and difficult in swallowing. You can read more up on this disease, and it is called goiter, right? That is by a lack of iodine in the diet, which causes a malfunctioning of the thyroid gland. Okay, I'm going to quickly look at the negative feedback mechanism of glucose. So here we have the normal glucose levels, right? When the glucose level in your blood increases, then the pancreas will secrete the hormone insulin. What does insulin do? It converts glucose to glycogen, which is stored in the liver, right? And the glucose level then decreases. Remember, glucose is now removed from the blood. It's gone into an insoluble form. It's glycogen now in the liver, and the level is brought back to normal. When you are fasting, for example, when you haven't eaten the whole day and your glucose level has decreased, then your pancreas secretes the hormone glucagon, right? What does glucagon do? It converts glycogen into glucose, right? Remember, you had stored glycogen. That stored glycogen is now converted into glucose by the hormone glucagon, which brings the glucose level back up to normal. Guys, you need to know the difference between glycogen and glucose. You need to know the difference between glucagon and insulin. And don't mix up glucose, glycogen, and glucagon. As I told you before previously, one letter is off. We're going to think that you don't know what you're talking about, you're not sure, and we're going to mark it wrong. So learn the spelling correctly. Right, thermoregulation. This is our last section. Thermoregulation is controlling the temperature in your body. It's a homeostatic response. It's homeostasis. How do we maintain our internal balance in your body when it is a hot day? Right? It's very, very hot. Like you're finding now, we're getting into summer. What's going to happen? And what's going to happen to your body on a hot day? Right. What happens is your blood vessels dilate. Right? That is called vasodilation, meaning vaso to do with blood vessels. They dilate, right? And that's the word we want. We want the word dilate. More blood flows to the skin, right? More blood flows to the surface of the skin. Remember that blood vessels do not move, right? Blood vessels do not move to the surface of the skin. And in many exams, we find learners writing this. It's more blood that flows to the surface of the skin, and heat is lost through the skin. Also, the sweat glands become more active and more sweat is released. We look for the words more because whether it is summer or winter, we perspire, right? Even in winter, if you're wearing too much of clothes and you take out your clothes, and you will find that your underarm, that you have been perspiring. So you need to understand that we sweat lots of times in the day, but during summer or when it is hot, more sweat is secreted. The sweat glands become more active, more heat is lost. Guys, please use the word more. When we talk about a cold day, right, the blood vessels constrict, and we do not mark the word contract correct because blood vessels cannot contract, they constrict. So make sure you use correct terminology, right? Blood vessels constrict, less blood flows to the surface of the skin, there is less sweating, and that's how we manage to bring homeostasis or a balance in the body. Right. This activity here is the homeostatic control of water. And let's look. The diagram below represents the homeostatic control of water levels in the blood when a person is dehydrated during a soccer match. Let's look at the diagram. There's a normal level of water in the blood here, right? Here, water level in the blood drops because remember we said this person is now dehydrated. So the water level in the blood is going to drop. Part A of the brain is stimulated. Which part of the brain I told you is responsible for water control, osmoregulation in the blood? Identify that part A. It is the hypothalamus, which is the temperature control, right? Then gland B releases more hormone C into the blood. What is gland B? Gland B is your pituitary gland. Remember, your pituitary gland is then going to release more of hormone C, which is your antidiuretic hormone, right? And your antidiuretic hormone is going to affect part D in your kidneys, which is your collecting duct and your renal tubules, okay? It will affect that, and that it'll bring the levels back to normal, okay? Your water level will then, if you are dehydrated, so what happens is you're going to have more antidiuretic hormone, which is going to make your renal tubules less permeable. So more water is going to make your more antidiuretic hormone is going to cause your tubules to be more permeable. So more water is going to leave the collecting ducts and go into your blood, and thereby maintaining homeostasis and preventing the dehydration from occurring. So remember, if it is more ADH, it's more permeable. More permeable means more water leaves the medulla, the collecting ducts, and goes into the medulla of the kidney and then goes back into your blood, and thereby ensuring that there is homeostasis and the person is not dehydrated anymore. State the effect of hormone C on structure D in this person. It becomes more permeable, as I said. Name one physiological process that will result in low levels of water in the blood of the person. That is the sweating. Right. Okay, now I'm doing one last activity with you. And I know I'm rushing, but we are pressed for time and we want to try and give you all the information that you require for this exam. Right? This one is an important one, so let's look at it quickly. The diagram below shows the structure involved in thermoregulation of the skin. Identify part A. Part A is your blood vessels, right? Explain the condition of part A on a cold day. So on a cold day, part A or the blood vessels constricts. Again, you need to use the word constricts, or you could say for the same mark, vasoconstriction occurs, causing less blood to flow to the surface of the skin. Therefore, less heat is lost to the environment. Guys, I have to stop here. Make sure you go over all of this. Have a comfort break. My colleague Radine Kruger will take over now for another important half that is coming up next. So Radine will do plant and human responses to the environment, another section that was answered very poorly during the prelims. Thank you very much.
Good afternoon, grade 12 learners. Welcome back to our life sciences lesson. My name is Radine Kruger, and I'm going to take you through plant and human responses to the environment. Okay, so we're going to have a look what the exam guideline says as far as plant responses is concerned. You need to know the functions of the 3 hormones, which is auxins, gibberellins, and abscisic acid. You need to know the role of auxins in phototropism and geotropism. And you also need to know how plants defend themselves against herbivores, so their protective mechanisms. And all of this is for 13 marks in the exam. Okay, so just to save a bit of time, I'm going to talk very quickly about the hormones, and I'm going to focus on the exam question that we had in the prelims, which was very poorly answered. Okay, so the first thing is, what are plant hormones? Well, they are growth substances, because remember, I don't know if you've ever seen a plant, if it's in the shade and it wants sunlight, it pulls up its roots and it walks over to find a sunny spot. Plants are not able to do this. So plants then have to grow. So their movement is a lot slower, but it's a growth movement. And they then grow in response to a stimulus. Now, it can either be towards a stimulus or away from a stimulus, or it can just be any or other growth movement. All right, so, and those growth movements then are controlled by what we call growth substances or plant hormones. Okay, and what they basically do is they control when a plant flowers, when does it, or it grows towards light, or it produces fruit, or when do seeds start to germinate, and that kind of thing. All right, so the first one we're going to have a look at is auxins. And remember, auxins is very important in tropic movement. So it is what regulates, or those auxins then regulate tropic movement. And so what they do is, auxins stimulate cell division. So in other words, the plant grows, as well as elongation of the cells. And once and if you have elongation that is uneven, so one side, the cells elongate more than the other side, what happens is you're going to have a bending of the plant, either the stem or the root, and that would be then in response to the stimulus. Okay, so that's the main function. And then because we know the functions of auxins, we have now when we understand them, we can now use them in agriculture. So very often auxins are used as a herbicide. And that is because when you spray an auxin-based substance on leaves, if they are broad leaves, which usually is what weeds are normally broad leaves, they're not long, thin leaves. So it doesn't have an effect on long, strappy leaves. So this is why we can use it. We can spray it on plants and it doesn't affect. We can use it on our grass, on our lawn. We can use it on maize products, because maize also has long, strappy leaves. And because of that, it doesn't affect the crops that we are trying to protect, but it does kill the weeds. And then the other thing about using natural substances, because this is a plant substance, is that it's not harmful to humans and to animals. So we're not polluting the environment by using plant hormones or auxins as a herbicide. All right, now, as I said, tropic movement or tropism is a growth response in response to a stimulus, and usually it's a directional growth. So in other words, it will grow towards the stimulus or grow away from the stimulus. Now, the 2 types of tropic movement that you need to know is phototropism, and photo means light. Okay, so it would be the response to light. And then geo is for gravity. Okay, so those are the 2 stimuli that we find in the environment that are then going to determine how the plant responds. Okay, so I'm sorry, let's have a look. Now, sometimes a tropic response can be positive. So in other words, when a plant, when the stem grows towards the light, we call that a positive tropic movement. And when it grows away from the stimulus, then that we call it a negative stimulus. And this is the one that threw us in the prelim exam. Okay, so let's have a look at that question. Now, the question that, or the stem of the question, and that's where you've got to read very carefully. You can't just go straight to the questions, because if you go straight to the questions, you're going to miss some of the information. And here, what they say to us, they said a pot plant was placed on its side. Fine. So we know if it's placed on its side, the stem is like this, and if it's going to, we want the leaves facing the sunlight, it's going to grow upwards. But they gave us some more information here, and they said it was placed in a dark box. So straight away, the stimulus could not be light. So we are not talking about phototropism. And that is what so many learners missed when they answered this question. Then they say, after 2 weeks, the stem had started to grow upwards, even though it was placed in a dark box. And when it's in a dark box, there's no light. Okay, but the fact that the stem had grown up. All right, so now the questions they asked us. The first one is they asked us to name the environmental factor responsible. So it's not the growth movement. Remember, the environmental factor is that thing that we find in the environment that is the stimulus or causes the change in the plant. Okay, so the answer for this one, and because it was in the dark box, it couldn't be light. Okay, so it had to be gravity. Right, so that was the environmental factor. Then secondly, they ask us to identify the phenomenon indicated by the stem's upward growth. So the fact that the stem grew upwards and gravity was the environmental stimulus. Remember, I said sometimes tropic movements can be negative. So what this actually shows us is it's geotropism, because there's no light. So it can't be phototropism. It is geotropism, but it's the negative side of geotropism, because the stem grows away from gravity, because light is not where gravity is. Remember, gravity is down in the earth, the center of the earth. So the stem doesn't want to go down into the soil because there's no light there. Okay, and light is very important for photosynthesis, as you know. Okay, so the phenomenon then is geotropism. All right, then the third question they asked us is name the plant hormone that is responsible. What caused that stem to grow upwards? And remember, we learned that it is auxins that regulate tropic movement. All right, so it's auxins. Right, then the next question they asked us is to describe how the distribution of the hormone mentioned in question 3.2 caused the stem to bend upwards. So in other words, what caused that upward growth, or how did auxins cause that upward growth? Okay, so we need to describe, they've asked us to describe it, and they've asked us to not just describe how, they asked us for the distribution. So we need to say where the auxins are found in the stem. Okay, so what we need to say is that we'd find more hormone or more auxins, or the auxins accumulated, or there was a higher concentration on the lower part of the stem. Okay, so the lower part, when the plant is lying on its side, we're going to have a higher concentration on this side of the stem. All right, so what happens then is that it causes more growth. So on that lower side of the stem, we have more growth happening. Okay, so those cells, there's more cells forming and there's elongation taking place. Whereas on the other side, where there's no or very little auxin, we have slower growth and slower elongation. So what happens then is it causes the stem to bend. Okay, so it causes more growth on that side, and we find fewer hormones or auxins accumulated on the upper part of the stem, and therefore there's less growth there, and that then causes the stem then to bend upwards. Okay. Then the last question they asked us was to explain one way in which the stem's upward growth benefits the plant. So here we can see, even though there was no light, the stem still grew upwards, because that is what a plant needs to do. And there's reasons why it needs to do that. And that is what you needed to actually explain here. So what you needed to say is that the leaves of the stem must be held up in the best possible position so that they can receive the most amounts of sunlight. Okay, so that is why, because if it's lying like this and the leaves are facing here, the light is here, then they're not getting the most, they're not making the most of the amount of sunlight that they could get. All right, and that is because of photosynthesis. Or there's another reason. The fact that when plants, grow upwards in the light, or the stem grows upwards, the flowers form, and they are also held in an upward position. And that makes them more favorable for insects that come in, that are flying in the air, to either pollinate or otherwise also for seed dispersal. So there are, once they form fruit and things like that, then the animals would, or even wind, could disperse. Okay, so those are the advantages. All right, then I'm just quickly going to go through the other two hormones, and that is gibberellins and abscisic acid. And just very quickly, the role of gibberellins is they encourage stem elongation. So in other words, we can also use them to cause stems to grow longer. They're also responsible for seed germination. So remember, seeds are formed at the end of summer, but then it becomes winter, and it's not ideal conditions. So why don't they grow in winter? They stay dormant, and only in the spring do they start to germinate, and that is because of the hormone gibberellins. They also promote flowering in plants. Then abscisic acid is the opposite. It is an inhibitor of growth. And this is very important that there is abscisic acid, because as the temperature decreases or it gets colder, the sun is further away, and the shorter daylight hours, there's less water, there's less sunlight for food. So the plant would not survive through the whole of winter if it still carried on functioning at the level like it does in summer. And also the fact that there's a lack of water. Winter is dry, so there's a lack of water, and that then also causes the stomata, which we found on the leaves. They go from open, where they can lose and give off water vapor, they now become closed. So that also reduces the amount of water loss. All right. There are investigations, and you may get these in the exam, and basically the importance of these is that you have a look and see. They can ask you what is the difference between the experiment and the control. Sorry.
You have been given power to decide whether you want good results in your life, in your schoolwork, or you want bad results. As Tony Robbins said, it is in the moment of your decision that your destiny is shaped. And your destiny is shaped right now with the decisions that you are currently making in your schoolwork. So decide whether you want to be an A student and start working on it, because there is power inside of you. The infinite power is inside of you, and the time you decide to tap in that power, you will become who you want to become in your schoolwork.
Okay, sorry, let's have a look again. So when we may get a question on an investigation, either for phototropism, or we could get one for geotropism. And the important thing here is to know, or to understand, the difference between the experiment and the control. Now, usually in an investigation, we're trying to test whether a variable has the effect that we think it's going to have on whatever. So in this case, we're saying there's a unilateral light. Will it cause, if there's a light from one side, will it cause the stem to grow towards the light? Now, in the experiment, we need to make sure that that is the factor that we are testing. However, to make our results more reliable and to increase our validity of our investigation, we very often introduce another set of apparatus, which we call the control. And what we do in the control is we eliminate the factor that we are testing. So in other words, in this case, even though this box has a single hole and there's light only coming in from one side, what we find is that this plant is on a clinostat. Now, a clinostat is a set of apparatus that turns. It turns very slowly, not very quickly, but it turns. So what happens is it'll keep that plant rotating in the box so that although there's only light coming in from one side, that plant keeps turning around. So it will be receiving light from all angles. And that is the difference between the experiment and the control. All right. And there you can see the results. In this one, the plant has grown towards the light. And in this one, even though the light is from one side, the plant did receive light all round and therefore continued to grow straight up. Okay, and as I said, you could also get an investigation on geotropism. And the same thing, if we have a wound clinostat, it means it's going to turn. And here we can test geotropism, actually, and we can see then the seedlings then will grow. So if they're constantly moving, so they're getting the stimulus from all sides, they're going to continue growing the way that they should. Okay, as you can see. Whereas if I have seedlings and I place them at different angles on the clinostat, you can see all of them, all the roots are growing downwards. Okay, so all the little shoots are growing downwards and the plumules, they all grow upwards. So in other words, they're going to grow towards the stimulus that is suited. All right. Okay. So the important thing to remember is where the auxins work in the stem. Okay, so we usually find auxins when there's a unilateral light. They are on the dark side or the side opposite to the stimulus. And there they stimulate growth, whereas in a shoot or the root, what happens is the auxins will then be accumulate on the lower side of the roots or the stem. So in other words, the side facing the stimulus, and they inhibit. And inhibit means to slow down or stop. Okay, so then it would have the effect of causing growth or a bending movement, growth movement. All right. Okay, so the difference between phototropism and geotropism, it's very good to learn it in a table like this so that you understand the differences. So phototropism is when the stem is exposed to a unilateral light from only one side, whereas in geotropism, when the root is placed horizontally. So it's not going to carry on growing horizontally, it's going to grow downwards towards gravity, because what is down there? It is water, and it's the root's function is to absorb water for the plant so that the rest of the functions can occur. Right, as I said, in phototropism, the auxins then are concentrated on the dark or the side opposite to the stimulus, to the light, whereas in the shoot, in geotropism, the auxin concentration is highest on the lower side, okay, which is actually facing the stimulus. So in phototropism, more growth is going to occur on the side away from the stimulus. So if the light's here, more growth is going to happen. And because those cells grow quicker and elongate quicker, it's going to cause a bending towards the light. In the roots, okay, what happens is it's on the lower side and it inhibits growth. So the cells above, on the opposite side, they are going to grow quicker because it's slowed down the growth and the elongation here. So what happens then? It's going to grow and bend and grow downwards. Okay, so that is the difference between the two. If you learn that, it's very easy then. Right, and then just quickly we're going to talk about the role of chemicals and thorns. So plants are very clever. Okay, they protect themselves from being eaten by herbivores. They have thorns. As you can see here, there's quite sharp thorns. Some of you will know those thorns of the acacia. They're long white thorns like that people have even can use even to sew because they are so strong and sharp. And then, of course, chemical defenses that plants produce that makes their leaves taste bad so that animals don't eat them. For example, tannins, which we find in tea, caffeine, all of those kind of things that we will find in a plant. Its purpose is basically to make the plant taste bad and therefore discourage the herbivores from eating them. All right, we now need to have a look at human responses to the environment, and quickly going to have a look at what the guideline says. So you need to know the brain. You need to know the following, very basically the following 5 structures. You need to know the basic structure and the functions. You need to know what the structure of the spinal cord. You must also be able to describe a reflex action using the diagram. You need to know about the different types of neurons that we find. And in grade 12, we focus on the functioning of the neurons and not so much their structure. All right, and then when we look at the eye, you need to know the structure of the eye. You must be able to label it. You should be able to give functions of those different parts. And then 2 very important processes, which is accommodation, which then allows me to look far away and close up. And then also pupillary mechanism, which is the where my eyes regulate the amount of light that is going into my eye so that my vision is good. All right, and then the last thing you need to look at, or the last section in this topic, is the ear. And there we again need to know the structure and the basic functions of all the parts, as well as the process of hearing and also balance. Okay, and then obviously we need to know some of the defects. All right, and this section is a very important section because it's out of 150 marks of the paper, it is 54 marks, which is a third of the paper. So if you can do well in this section, then you should be able to do well in your paper. All right, so the first thing we're going to have a look at is how our nervous system works. And remember earlier, Fauzi said that the endocrine system is a lot slower, whereas the nervous system works quickly. Okay, and basically what it does is we have sensory receptors, or we have sense organs that allow us to respond to stimuli in the environment. So remember I said to you with plants, they are also able to respond to things that happen in the environment. The same thing with humans. So there's a stimulus which is picked up by a receptor. For example, our eyes, the stimulus is light, and then we have receptors, photoreceptors, which then are receptive to that stimulus. And that then triggers an impulse, which is carried via our sensory neuron. Remember, sense, for our sense organs, is a sensory neuron. So that's going to gather information from the environment, and it's going to take the impulse and that information to the central nervous system. Okay, so the central nervous system is your controlling structure. So in other words, it's going to get the information coming in, it's going to analyze it, and it's going to say, okay, this is how the body needs to respond to this stimulus. And then it's going to send out an impulse via the motor neuron. Now remember, M for motor, M for movement, because very often if there's too much light, if the light's too bright, my pupils are going to constrict. And even if it's so bright, I'm going to squint or possibly even close my eyes. Okay, so the response goes along the motor pathway. And remember, M, motor, for movement, if you like. Okay, we said sensory for sense organs. Okay, so the S. So that's how we remember the difference between the two. Right, and your motor neurons then are going to carry the impulse to an effector. And an effector is something that's going to bring about the reaction to whatever the stimulus is. All right, so, and that will then be your response. Okay, so that's how your nervous system works. And if we have a look at the nervous system, it's divided into different parts. There's 2 main parts, and the first is the central nervous system, and that consists of the brain and the spinal cord. And then we have another section which is called the peripheral nervous system. Now remember, the periphery means the boundary, the outer area. So your central nervous system is your brain and your spinal cord, and your peripheral nervous system is everything found on the outside of that. So for example, like at school, the periphery of your school grounds would be the wall or the fence or whatever it is that you have surrounding your school. Okay, so the peripheral nervous system is what branches off the brain and the spinal cord. And we call those cranial nerves, and we call them spinal nerves. Right, now our peripheral nervous system, as I said earlier, has 2 pathways. The one is sensory, And sensory is going to bring the information in because of from the sense organs, which are sensitive to stimuli in the environment. And then there's another pathway called the motor pathway, so that once the central nervous system has integrated and analyzed that information, it's going to bring about you need to respond. So then it's going to send an impulse via the motor pathway to bring about that response. Then if we have a look at our motor pathway, it's also divided into two parts, and that is the somatic. And somatic, remember, somatic cell means body cell. So those are all the nerves that go to the different parts of your body and usually control our voluntary actions. So in other words, me moving my hands, walking from here to there, sitting down, whatever. So that is my somatic nervous system. And then I have the autonomic nervous system. And the autonomic nervous system, as it says, it works automatically. So in other words, when somebody shines a bright light right here in my eyes, my pupils, I don't have to think about my pupils closing down, because if they don't, my retina is going to be damaged. So in other words, your autonomic responses are usually in response to a harmful or a dangerous situation. And that, if we have a look there, it also has two parts. One part is a sympathetic, which is going to increase your ability to respond to that harmful situation. And the parasympathetic, because I can't function at that level all the time, is going to slow down my body. All right, so let's have a look at a question. Oh no, we'll first have a look at the different parts of the brain. As I said, you need to know the basic structure and the functions. So the cerebrum, that is the largest part of your brain. So it's this part here. Some of the functions is voluntary action. So it's the part that allows me to speak, to hear, to move, whatever. And then also it helps you with memory. It's also my intellect, when I'm studying, where do I store the information? It's in my cerebrum. So that is this part of your brain. Okay. Then we're going to have a look at, we know that the brain has actually two hemispheres, two halves, and there's a structure, so we can't have them separate. They must be joined together. And that structure that joins them and works almost like a bridge that allows information to go from one side to the other is known as the corpus callosum. And remember, you learned when you did human reproduction that corpus means body. Okay, so it means, and callosum is white body. So in other words, it's a white layer. So this part here, this is quite, if you look at it compared to the other parts of the brain, it's quite white. And that is why it's called the corpus callosum. And it just basically joins the two halves together. Right. Then joined to the brain is the pituitary gland, which is very important. It works along with the hypothalamus and it regulates all the functions in our body. But the pituitary gland is a hormonal gland. All right. And then the next part we're going to have a look at is down here, what we call the brain stem. So it's the part that joins to the spinal cord, the medulla oblongata. And the medulla oblongata is very important because that's what keeps you alive. Okay, so if you damage it, it's going to affect your breathing, it's going to affect your heartbeat, it's also going to affect impulses that go to the rest of your body. All right, and then the next part is the cerebellum. It's the part at the back here. And the cerebellum, when we learn about the ear, we'll learn that our balance and equilibrium, that is controlled by the cerebellum. And then the last part of the brain is called the hypothalamus. Okay, and the hypothalamus is your control center. And that's why the pituitary is joined to your hypothalamus. So it's your control center for hunger, thirst, emotions, temperature, tiredness, yeah, all of those. All right, as I said, the peripheral nervous system, we said, is that branches off, and it's very important that it then carries either information, because remember, sensory is going to come into the central nervous system and motor is going to go away from it. So it regulates and it ensures that we are responding to changes in the environment all the time to make sure that our bodies function at the best level possible. All right. Okay, so this was a question that I was talking about. It comes from the Mpumalanga prelim paper 1. And here they say John has to deliver a speech and he's feeling extremely nervous. This may be due to an autonomic reaction or automatic reaction to a stressful or frightening event brought about by a branch of the nervous system. So the first question they ask us to name that system. And remember I said to you, our autonomic nervous system has two parts. Now, the one part that's going to lift you up so that you can react to that harmful or stressful situation is our sympathetic nervous system. So that would be the answer for that question. Right, then they ask us to provide 4 symptoms. Now, remember you learned about adrenaline, and you learned that adrenaline, when your body has adrenaline, it stimulates your heart, it increases your breathing rate, all of those things. Now, it works exactly the same as your sympathetic nervous system. But remember we said nervous system, the impulses travel very quickly. So they work a lot quicker than the endocrine. So imagine you standing in the middle of the road and there's a car coming towards you. And your body now has to wait for the adrenal glands to release adrenaline and for the adrenaline to get all around your body in the blood. And only then, once it gets it, I jump out of the way. Probably the car would have run me over by then. So this is the sympathetic nervous system works a lot quicker. So it's first going to kick in and then adrenaline is going to take over. Okay, so what it causes is it causes increased heartbeats. And remember, when your heart beats faster, blood moves around your body much quicker, so your breathing rate is going to increase. It also leads to dilated pupils because remember, I need to be able to see clearly and respond. It also takes all the blood away from your skin because we don't need the blood there. We need them at your muscles and your heart and your lungs and your brain. So it gives you a pale complexion. It might cause shivering. It also causes dry mouth. I'm sure all of you have experienced that. You also have sweating that can occur. And then the feeling of the constant need to urinate. And all of those then would be the symptoms of your sympathetic nervous system. All right. Then, now, that is what Fawzi said to you. Remember, learn that adrenaline and your sympathetic nervous system. Examiners love to put them together. So yeah, they've said, name the hormone and gland that work with the peripheral system during stressful situations. And that would be, the hormone is adrenaline and it comes from the adrenal gland. All right, then they say, suggest a strategy he can use to physically calm himself down. So that's what we normally do when people get all upset and excited and hyper. Then we say to them, calm down, take deep, slow breaths. And maybe also, contract and relax your muscles so that they then start to slow down. All right. Okay, now what's important in grade 12 is that you know the functioning of the 3 different types of neurons. And here we have a motor neuron, and remember motor, and it's a multipolar neuron, so there's many dendrites in a motor neuron, whereas a sensory neuron has a single dendrite, so it only has one dendrite, okay? And the interneuron is the one that we're going to find that connects between a sensory neuron and a motor neuron. Okay, so learn those, learn the structure. You may be given a diagram, and that's what we're going to have a look at, an exam question now. This one came out of the Eastern Cape paper, and they gave them 2 diagrams, 2 different neurons, and they needed to answer questions. So the first one is they asked them to identify the neuron in diagram 1. We can see it's multipolar. Okay, so it has many dendrites, and therefore it would be a motor neuron. Whereas this one has only one dendrite, or mostly, that goes into the cell body. So therefore it is a sensory neuron. All right. Then they say, identify part C. So here they're asking you about the structure of the cell, the neuron, which is a nerve cell. So you need to know the structure, and that would be the cell body. And then they say, give the letter and the name of the part that provides electrical insulation. And remember that the extensions very often are wrapped with a fatty layer. It's almost like a Swiss roll wrapped around the axon or the dendrite. And that is insulating, and that is known as the myelin sheath. And if we have a look here, this label here, they've labeled myelin sheath. And then they say the letter and the name that receives incoming impulses, and that would be the dendrites, or A, they've labeled them here. So incoming impulses come into the dendrite, into the cell body, and then out along the axon to the next neuron. And then the last question they ask is, which neuron, one or two, transmits impulses to the central nervous system? And remember, we said sensory goes to, motor goes away. So it would be diagram 2, or the neuron in diagram 2. All right. Okay, now what is very important in this section is you must know the difference. You must know the terminology. There's a difference between a reflex action and a reflex arc. Now, a reflex action is a very quick reaction, okay, automatic response to a stimulus. The plate on the stove is hot. I don't know that it's switched on, and I happen to put my hand and I pull it away very quickly before I burn myself any further. That is a reaction. The pathway in which the stimulus went from the sensory cells in my skin, on my hand, up my arm, to my spinal cord, integrated, back down through the motor pathway and told the muscles to contract and pull my hand away. That's the actual pathway, and that is your reflex arc. So it's very important that you know the difference between a reflex action and a reflex arc. Okay, and then always they say, don't ever talk about it sending a message. The sensory neuron doesn't send a message. It carries an impulse. Okay, you won't get marks if you write message. Right, then if we look at a diagram of the reflex arc, as I said, so there the impulse is going to travel along the sensory pathway until it gets into my spinal cord, where it connects with the interneuron. It's going to integrate the information, and then it's going to send out an impulse to my effector, and in this case it is the muscle, to pull my hand away from the structure that's going to injure me. Right, now the questions, this was in the Free State paper, and here they've given us a very nice diagram of the spinal cord and a reflex arc, and there they've then labeled certain structures. So the first question they ask is, what is a reflex arc? And I remember we said the arc is the pathway. So in other words, it's a pathway along which nerve impulses are carried from the receptor to an effector to bring about a reflex action. Then the next question they ask us to explain the effect on the reflex action if or when neuron C is damaged. So in other words, this one is damaged here. So what happens is the stimulus will still, the impulse will still be carried in from via the sensory neuron into the spinal cord, but because there's damage to the neuron that goes to the effector, the person won't be able to react. So let's have a look what they say. A person would be able to feel the sensation. So they would feel whatever it is, feel the pain or the burning or whatever, but it's unable to react to the stimulus. All right. So then they ask us to name the disorder that results in the breakdown of the myelin sheath of the neurons, and that is known as multiple sclerosis. All right, let's quickly have a look at the eye. As you can see, you need to know the basic structure of the eye. You'll need to know the outer layer is known as the sclera, the middle layer is known as the choroid, and the inner layer is the retina. Now, the most important, or what the eye's function is to be able to see. So if we have a look at the front part of the eye, so although the sclera is white and opaque, which means we can't see through it, the front part of the eye is transparent, and it must be transparent to allow light in. But you can also see how nicely curved that front section is, isn't it? Flat. Okay, so the more curved it is, the more it's going to refract light. Okay, and it's important that light is refracted. So remember, light travels in straight lines. But to see a clear image, the light must be bent so that it converges at a point on the retina. And that is what then gives us a clear image and not blurred vision. All right, so the parts of the eye that are going to refract would be this part here, which is known as your cornea, as well as this structure in the middle here, which is the lens. Okay, and then inside your eye there is obviously fluid. There's a very, at the back there's a jelly-like layer or fluid. It's not so runny, it's quite jelly-like. And then in the front of your eye there's a more watery fluid. And that then gives your eye shape. Because if you don't have shape in your eye, if it loses turgidity or shape, then you're not going to be able to see very clearly, because then the structures need to be in a certain place and they need to be rounded so that they can refract light. Then at the back of the eye, this is where your retina, the inner layer of your eye, and that is what has your photoreceptors, which are those that are sensitive to light. And then we have a spot at the back of the retina, which is known as your fovea or your yellow spot, and that is where your most precise vision. So if I want to look at something and look at the fine detail, then I hold it close up and the light strikes there and I can see the fine print on something. We also have a part in the retina or the back of the eye where there are no photoreceptors, and that is because the nerve leaves the eye, and that is known as your blind spot. So if light falls there, then you won't be able to see. And you've heard, and those of you who are perhaps driving or learning to drive, you know that people say to check your blind spot, and this is why you have those two side mirrors to help you check your blind spot, because you wouldn't be able to see if there's something, another vehicle in your blind spot. All right. Okay, so the first function that we're going to learn about is accommodation. And accommodation is when my eyes adjust so that I can see far away and I can see closer. So let's look at the first. So when an object is less than 6 meters away, very important there, we have ciliary muscle. And the ciliary muscle is a structure that's going to contract. And the muscle fibers run like this. It's a circular structure. And when it contracts, it then makes the space, the lumen inside, smaller. And when the ciliary muscle contracts, your sclera is pulled forward, your suspensory ligaments, that are the ligaments attached to the lens, they slacken. So they no longer pull tight. So they slacken. And when that happens, so the suspensory ligaments loosen or they slacken, tension on the lens, so they're no longer pulling on the lens. So that decreases. And as soon as that tension decreases, the lens becomes more convex. Okay, so if it's the suspensory ligaments are tight, it would be flatter, but when the tension decreases, then the lens becomes more convex. Okay, so please remember we've put in a little mnemonic here for you to remember. A ciliary muscle contracts, suspensory ligaments slacken, tension decreases, lens becomes more convex. It's going to be able to refract better. Right, now when an object is further away, the ciliary muscles relax. So now that ciliary muscle that was contracted now relaxes. And as soon as it does that, the space in between now increases. And when that happens, remember the suspensory ligaments are attached to the lens and then to the ciliary body, then the tension increases. So in other words, my suspensory ligaments then become tight or taut. And when that happens, they're pulling on the lens. So the tension on the lens then increases. And when that happens, my lens now goes from being nice, fat and round, it now becomes thinner and longer, so it becomes more elongated and flatter. All right, so it then becomes less convex, and that decreases the refractive power. And again, our little mnemonic there is ciliary muscle relaxes, suspensory ligaments become tight, taut. Remember, they can't relax, our ligaments. The tension then increases on the lens, and the lens becomes less convex. And so then I'm able to see on whatever the object is further away. Right, and here you can just see the difference. So here, the suspensory ligaments are taut, tight, and therefore it pulls on the lens and it makes it longer and thinner. Whereas in this case here, my suspensory ligaments are slackened or loose, and so my lens becomes rounder. When my lens is rounder, my refractive power increases. When my lens is elongated, longer and thinner, my refractive power decreases. All right. Okay, then a question in the Free State paper, and if we have a look here quickly, they say the diagram represents part of the eye responsible for accommodation. This is the front view, so here you can see the ciliary muscle. Here you see the little suspensory ligaments, and they look quite loose, and then that obviously is the lens. Okay, and that's the side view. Now they say the following statements are used to describe accommodation in the eye. Ciliary muscles contract, suspensory ligaments become taut, tension on the lens increases, lens becomes more convex, refractive power of the lens increases. So let's have a look at which ones are relevant to this diagram. So definitely the ciliary muscle has contracted, because we said when it contracts, the suspensory ligaments loosen, they become slack. So it's not this one, because it says they become tight or taut. Tension on the lens increases, so that's correct. The lens becomes more convex, and the refractive power, let me just see, yes, becomes more convex and the refractive power increases. So the correct answer there, which of the statements relate to this diagram? And let's have a look. So if we've got, it's number 1, 3, 4, and 5. So let's have a look. Yeah, it's 1, 2. Okay, so it's actually only 1, 4, and 5. Tension on the lens. Tension on the lens doesn't increase, it decreases. Sorry. Okay, so then the correct answer would be C. Okay, so let's see. Yes. All right. Pupillary mechanism. Okay, and that is where your pupil regulates the amount of light that is entering the eye, and it's very important because if you allow too much light into your eye, it will burn the retina. So in bright light, what happens is we have circular muscle and radial muscle. So the circular muscle contracts, and when the circular muscle contracts, remember we said the space becomes smaller. The radial muscle must relax, because if it doesn't work opposite, then we won't have any change, any movement happening. So your radial muscle relaxes, your pupil constricts, and it allows less light into the eye. Whereas in dim light, when I need to see, because now the light's very dim, so I need to allow more light in. So here my circular muscle now is going to relax. And when that happens, my radial muscle must contract, so it pulls it out, and it makes my pupil wider. So my pupil is going to dilate. And when it dilates, it's going to allow more light in. Okay, so use those mnemonics that when you get your paper, write them down quickly so that you remember. And then it's very easy to be able to describe the process. All right. I'm going to skip this because I'd like to go to this investigation question. And here, specifically, this was from the Free State prelim paper, and investigation questions are poorly answered. And here, if you have a look, remember in investigation, we always look for the aim. And here they say an experiment was set up to investigate the diameter of the pupil under different light conditions. Okay, so that is my aim. When they are going to show something, prove something, investigate something. And here they want to see the diameter of the pupil. How does it change? Under different light conditions. So there's my aim. And as soon as I've identified my aim, then I can go and get information from there. Further, they say a person sat in a dark room and an electric light bulb was switched on at different distances from the person. So they changed that. The diameter of the pupil was measured 1 minute after each time the light bulb was switched on. The results are illustrated in the graph below. Okay, now the examiners love to ask us this very first question. Name the following in the investigation: the independent variable and then the dependent variable. And this is why I said to you, it's very important to look for the aim in the stem of the question, because the aim will tell us which is the independent variable and which is the dependent. So in this case, in the aim, they tell us the diameter of the pupil is one of the variables and different light conditions is the other. Now, which one am I manipulating? I'm manipulating where the light is in relation to where the person is situated. So if I, the investigator, am manipulating that, that would then be the independent variable. Whereas the dependent variable is the one that is dependent on the independent variable. So that would be the other one. And that is the diameter of the pupil will change depending on where the light, how close or how far away the light is from the person. So my independent variable would be the different light conditions, and my dependent variable would be the diameter of the pupil. All right. The second question they ask us, state 2 reasons why. They don't like to ask us directly anymore, how can we increase the reliability? So they'll say to you, give 2 reasons why the results of this investigation are not reliable. So you have to go and look at the investigation to answer. You have to apply it to your case that they've given you, the case study. And in this case, they say that only one person was used. They didn't use a whole lot of people. Okay, so if they use more than quite a few people, increase their sample size. So if they use more people to participate in the experiment, or they increase the sample size, it would have made it more reliable. So therefore it's not. They also didn't say they did the experiment several times. They only did it once. Okay, so therefore the experiment, the results are not reliable. Right, then they ask you to calculate the percentage decrease in the pupil diameter when the light bulb was moved from position 3 to position 4. Now this is a favorite calculation question that we get in life sciences. They like to ask either percentage decrease or percentage increase. So we now need to work out from position 3 to 4. So if we look at position 3, we can see that it's 8 millimeters. And if we look at position 4, we see it is 5 millimeters. Okay, so let's have a look at what they say. Okay, so we say 8, so it's the starting number minus the end number. Okay, so the initial number minus the end number divided by the initial number. And then we multiply that by 100, and we should get our percentage increase or decrease. So in this case, they're asking us about a decrease. So they say that here you can see the pupil diameter decreased from position 3 to position 4 by 37.5%. Okay, and remember, even if you're not sure of what values to put here, if you say times 100, you will get a mark. Okay, so please remember that, even if you're not sure how to do the calculation. But it's very important or very easy to learn starting value, end value divided by the starting value. Okay, and that will give you your answer, multiplied by 100. All right. Okay, then basically the structure of the ear. We know that the ear has 3 parts, the outer ear, the middle ear, and the inner ear. And obviously all of them have a specific role in the function of hearing and balance. Okay, so very important, and I don't have time to go through all of it, but the middle ear and amplification of sound. They could ask you this question, and that is because the tympanic membrane, or your eardrum, is much larger than the oval window, which is attached to the stirrup so the 3 ossicles. And because you have a large surface that is vibrating, and those vibrations are transferred to the little bones, the ossicles, and it condenses it down into a much smaller membrane that is then going to vibrate. What we are doing is we are amplifying, or we're increasing the vibrations. Okay, so, and in this case, it amplifies it by 20 times. All right. Okay, so let's have a look at hearing. What's very important is, as we said, the sound waves are trapped by the pinna. They then go via the auditory canal to the eardrum. The eardrum vibrates, causes the vibrations of the little ossicles. So now we no longer have pressure waves or air waves. It's now vibrations, and those vibrations are then transferred to waves, fluid waves in the inner ear. And that's via the oval window. So it sets up waves in the fluid that we find in the cochlea, and when those waves occur, the membranes of where the organ of Corti is found move, and it causes the little hair cells to be triggered, and that then stimulates an impulse, which then goes via the auditory nerve to the cerebrum. Okay, and examiners love to ask this. From hearing, from the cochlea, it goes via the auditory nerve to the cerebrum. Okay. Right, so we see the path of sound. It's caught, and you can see, if you have a look at the size of the waves, these are sound waves, which then are converted in the ossicles into vibrations. And there's the amplification, where we've increased the size of the waves. And that then goes via the oval window into the fluid, which we find, and that then stimulates the organ of Corti, and the impulse then is carried along the auditory nerve to your brain, to the cerebrum. Right, now balance and equilibrium. Those are the parts, and this is the part that learners often battle with. But it's the different parts of this vestibular apparatus, and here we have the semicircular canals. Now, semicircular, they are sensitive to changes in speed and direction. So when I'm running around on the soccer field, on the netball field, rugby field, on a tennis court, just around the track, there's a difference in the speed and the direction that my head is traveling in, and those receptors in those structures then are sensitive to changes in speed and direction. Whereas the other receptors that are called maculae, so those are cristae, they're in the semicircular canals. The other ones that we find in the sacculus and utriculus are known as maculae, and they are sensitive to the position of my head. So as soon as I bend my head down, those receptors are stimulated, because now the direction of my head has changed. As soon as I change the direction of my head, those receptors are going to be stimulated. Right. So the movement, and what happens is, as soon as I move my head, or I'm running around and looking this way and then looking that way, and then soccer ball goes this way and then suddenly it goes back that way, I'm changing direction and speed all the time. So when that happens, there's fluid that is also there, and the movement of that fluid then causes those receptors to be stimulated, and they then trigger an impulse, and they then also, that impulse, that nerve impulse, is also carried via the auditory nerve, but this time it goes to the cerebellum, because remember we said the cerebellum is the part of your brain that is responsible for balance and equilibrium. All right. Okay. Right, so the two ways in which your ear regulates balance, and again we've got a little mnemonic here that hopefully helps you, remember this better. So for our semicircular canals, remember they are sensitive to direction and speed. This receptor is called a crista, and they are found in the ampulla, which is the base. So in other words, a semicircular canal, it has a swelling at the base, and that's known as the ampulla. And so the little mnemonic we have there is acids. So hopefully you're going to remember. Then the second part is your again, a little mnemonic, sump, where the receptor is sensitive to the position of the head towards gravity. Those are the receptors are called maculae, and they are found in the sacculus and matriculus. And they then, their function is to basically control balance and equilibrium. So when those receptors are stimulated, they then are going to initiate an impulse, a nerve impulse, and that impulse goes via the auditory nerve, as we said, but it goes to the cerebellum. And try and remember, cerebellum and balance sound similar. Okay, so the cerebellum is what regulates balance. Okay. All right. Here's quickly a question from the Eastern Cape prelim paper on the ear. And you can see it's the different parts, so we can basically see it's the inner ear. And they ask us to identify part D, which is the cochlea. State one function of E, so that is our auditory nerve, and that would be to carry nerve impulses or transmit nerve impulses to the brain. State why part A must absorb the excessive waves, that's the round window. Okay, so it's going to release, it's like a pressure valve, and that is so that we don't have echoing happening in our ears. Because if that fluid just keeps bouncing and we don't relieve the pressure, it's going to cause echoes. And then to explain why a rupture or hole in part B, so that would be our oval window, so if there's a hole there, it may lead to hearing loss, and that's because as soon as there's a hole, it's not going to vibrate. Okay, so if there's no vibrations, then no pressure waves will be caused or created in the inner ear, and if there's no pressure waves there, then our organ of Corti is not stimulated, and therefore we will have no impulses sent to the cerebrum, and if there's no impulses there, then we don't have any hearing. Okay, then they say, describe how the structures indicated by C maintain balance. Okay, and let's quickly look at that. Sudden changes in the speed and direction of the head stimulates the cristae in the semicircular canals, and a change in the position of the head would stimulate the maculae, which then is in the matriculus and sacculus, and that sends impulses via the auditory nerve to be interpreted at the cerebellum. Okay, thank you very much. That is all we have time for today. I wish you luck. Study hard. It's not just about luck, it's about working hard and maximizing all the resources that you have. Your teacher is there to support you. And then remember, very important things to take to the exam room so that you can answer your question is you must have a pen or a pencil. You also should have a ruler. You should have a compass. You should have a protractor, like we had to draw a pie chart now in paper 2, and then also a functioning calculator. Okay, so make sure you have all of that equipment, and then good luck for your exam. Thank you.